Welcome, science friends, to translation. Unlike transcription that is staying within the nucleic acid language, from DNA to RNA, they're both nucleic acids. Translation is a completely new language. We're going from nucleic acid language, the mRNA, into a different macromolecule, amino acids and protein language. So it gets a little bit more complicated. So how does mRNA code for proteins? Well, we see the DNA, great, that's the first step. Then we transcribe it into mRNA, but then something has to happen to go from that one macromolecule language to a different macromolecule language of protein. We have 4 different types of DNA nucleotides: adenine, thymine, cytosine, and guanine. We have 4 different types of nucleotides for RNA. We have adenine, uracil, cytosine, and guanine. But for proteins, there are 20 different amino acids. So a really big question is, how do we go from 4 to coding to 20 different things? What we look at with our translation process is that we look at our little codes in our DNA by threes or triplets. We see the 4 different nucleotides for DNA, we see 4 different nucleotides for mRNA, and we look at them 3 at a time, and those are called codons. Each codon gives us the directions to make one amino acid. Just like we translate between language, so from maybe English to Spanish or from Mandarin to English, we need to translate instead of making a copy. We need to change the language, which is what we're seeing here in translation. Once again, going from nucleic acid into protein language. The organelle of ribosomes help us, and they also have another new type of RNA called transfer RNA, or tRNA for short. So the ribosomes and the tRNA work together to help with this translation process. Each codon, or set of 3 nucleotides on the mRNA, is the directions for one amino acid. It's just like a code. And when we build up those amino acids, one at a time, we make chains, and those chains begin to fold and make our proteins. So the code is really, really important, and we see a lot of really cool things happening. The code is the code for all life. Without proteins, we can't have life. It is redundant as well, because there's a lot of different combinations of those 3 nucleotides, but there's only 20 amino acids. So here we can see that we have redundancies or repeats for many of our amino acids. Some important amino acids to point out are 2 of them. We have first the start codon, which says, hey, we're going to start making our protein right here. And that is methionine, MET for short. And the code for our codon would be AUG. So anytime you see AUG, it's for methionine, or MET for short. We also have another special set of codons called stop codons. And these tell our ribosome and our tRNA to, hey, stop. We're done with the protein, we don't need any more. And there are actually 3 different codons that tell us to stop: UGA, UAA, and UAG. So how do ribosomes translate that mRNA into protein? Ribosomes have helpers, so to speak, in a different form. So instead of mRNA, we're looking at that tRNA. It's called transfer RNA because it's transferring the code information into a protein information. tRNA matches the codons from the mRNA with an anticodon on its base. So it's on the bottom part of our tRNA, there are 3 nucleotides, and those 3 nucleotides must match the codon nucleotides. Anti just means the opposite, so it's the opposite of the codon on the mRNA. At the top of every tRNA is an amino acid that matches the code on the codon for the mRNA. While it's in the ribosome, we see the attachment of those amino acids to create an amino acid chain, also known as a polypeptide chain. The last little bit here, we are going to build a polypeptide, or a long chain of amino acids, and there are 3 steps. So in our ribosome, there are 3 channels where those tRNA will go and help build that polypeptide or that protein. The first step is initiation. We're bringing together that mRNA, the ribosome, and the tRNA with that start codon or the start amino acid of methionine to match the start codon of AUG on the mRNA strand. So we got to start the process of building our protein. The next step is called elongation. We are elongating or making longer that chain of amino acids that will become our protein. So all that means is, great, we started it with initiation, now we're going to make it longer and keep going with our chain during elongation. The last step is called termination, meaning we're going to terminate or end that protein chain. So basically we just run into one of the stop codons, which signals to our tRNA and the ribosome, hey, we've reached the end, there's no more amino acids, our chain is complete. Our chain will then move on into a folding process, usually within the endoplasmic reticulum, to fold up and become the shape that it needs to succeed. Here's a quick little animation from Amoeba Sisters showing us, hey, there's RNA polymerase making a copy of the DNA. That copy heads off to a ribosome where it comes into contact with our tRNA that have amino acids, and we're building the chain. So in summary, mRNA codons that we made during transcription are matched with complementary tRNA. Each codon, that set of 3 nucleotides, codes for one of 20 amino acids. There are lots of different combinations of those nucleotides, so we have redundancies or repeats of those codes for each amino acid. Important codes to know are the methionine or AUG start codon. We also have those stop codons, but the start codon is the most important. tRNA builds the protein 1 amino acid at a time in the ribosome. So we see it come in, it attaches the amino acid to make that chain, which will eventually become our protein. We have the 3 steps of the building: the initiation, elongation, and termination. The start, the growing, and the stop. What we are learning right now, all of the stuff about RNA, but mostly the transcription and translation processes, help us understand how life forms. Without proteins, we don't have life. So this process of we take DNA and we transcribe it into RNA and we translate that into protein is the basis of biology. This is called the central dogma of biology. A dogma is a principle or set of principles laid down by an authority. So like the big science people say is incontrovertibly true. So true without a doubt. This is a primary basis of biology. We can't change this. We can't throw it away. Without this process of protein synthesis, biology is nothing. So it's really important that we understand that this protein synthesis is the basis of all life. Without protein synthesis, we can't have life. So we call that the central dogma of biology. DNA makes RNA, RNA makes protein. That was translation. I hope it helped.